Hello, my name is Natalie Ford. I'm a postdoctoral researcher at the Donderst Institute in Nijmegen, and I work on the AIMS-2-TRIALS data. So within AIMS-2-TRIALS, I have two different roles. I'm working on LEAP, so it's the Longitudinal European Autism Project, and I'm the local coordinator for the LEAP 3-wave data collection. And I also work analyzing data from LEAP Wave 1 and 2. And this introductory video is going to be about me discussing some of the research I'm doing on that data and preparing you for the webinar in March. So I work with brain imaging data, and what I'm doing in particular is linking multiple sources of brain imaging data together. So many studies have, and much research has already been done on each of these different data types with some very interesting results, but unfortunately, there is not too much consistency across studies. Part of the reason for that is that there's a huge amount of variability within individuals, and of course, within the autism community as well. So often studies don't have the power to detect subtle differences, or the methods they're using when used alone don't have enough sensitivity to detect any potential brain behavior relationships. So that brings me to the approach that I'm using, and some of my colleagues are also using, which is then to integrate different data sources of information to try and get a higher sensitivity to detect any potential relationships between the brain patterns and behavioral patterns. Another thing that I've been doing in my research is, instead of just looking at what we call case control differences, or differences between the groups on average, we also look at variability across the whole sample. So within the autistic and neurotypical groups together. When we think about brain tissue, we normally classify it into two different types. We have gray matter and white matter. The gray matter is generally thought of as the processing hubs of where all of the information and things are happening. But the white matter is just as important, and it's what is connecting, the structural connections between these different regions that allows communication between regions, that facilitates the brain functioning at all. So one of the methods in particular, the diffusion weighted imaging, specifically we're using to look at how these connections or white matter structures between regions are organized. I'm using 3 different types of brain imaging data, and I think I can probably run through all those 3 to give you a bit of an introduction for the webinar. The first one that I use is what we call, it's a standard anatomical image, specifically called a T1 weighted image. And this is almost like a static picture of the brain. So you can identify the different tissue types, like the gray and white matter in the brain. And with this kind of image, we can see, or we can investigate differences or variability in the size or shape or the volume of brain regions. The second data type I use is diffusion weighted imaging. And this is sensitive to the microstructure of the tissue within the brain. This is particularly useful for looking at white matter tracks. And the white matter is the fibers that connect different distant regions of the brain. When we use diffusion imaging, there are multiple ways of analyzing the data or modeling the data to identify these white matter tracks. Traditionally, something called diffusion tensor imaging, or DTI, is used, but this method is quite limited as it's an oversimplification of the complexity of the white matter fibers. Instead, there are multiple methods, and we can use them to model the signal that we get from the MRI in a more representative way of what the brain organization actually is. In the image, you can see an example of what this looks like if we really zoom in and look at how it's modeled using a diffusion tensor image versus a more complex model. In the diffusion tensor image, you can see that even if there's crossing fibers, that's then aggregated to one ellipsoid. Whereas if you have a more complex model, like we're using in this, like I'm using in this research, you can actually identify different crossing fibers. The third data type I use is functional MRI, and we use a resting state scan. So the participants are asked to lie in the scanner and not do anything in particular, but just let their mind wander. There are many ways of analyzing the data, but traditionally, the data is analyzed in a way that regions are given one potential function and the signal is averaged or aggregated throughout that region. So the idea there is that that whole region is responsible for kind of the same thing, and that there's no smaller organizations within it. But we know that's not the case, and we have plenty of research that shows that the brain and different regions of the brain show multiple organizations, potentially show multiple organizations within one region and multiple functions. So they can be involved in various different things. So instead of using parcels to define brain regions, we use something called connect-topic mapping. And then we look at how a region is connected to the rest of the brain, but allowing it to be, to have a more fine-grained organization or a gradient within that region. So as you can see in this example image, traditionally you would have had, the region would have been one organization, one color, let's say, and instead we have these multiple organizations within the region that are shown by these gradients, these rainbow colors. The next step then is to integrate all these different data types. So I take the anatomical images, the diffusion images, which is the structural connectivity, and the functional images, which are these gradients of functional connectivity. And we use a method called linked independent component analysis. And this basically looks for patterns of variance that are shared across the three different data types. And then reduces it down to a smaller number of components for us to investigate with our statistics. For the statistics, we use quite simple linear models to investigate the association between various clinical measures or behavioral measures and the brain patterns that we just extracted using linked ICA. There are multiple behavioral measures that we look at that are relevant to autism. These include things like social communication, repetitive behavioral skills, and measures of daily functioning, so general daily functioning. For a lot of these, we use structured interviews, like the ADOS, to get measures of some of these things. And the Vineland is also a structured interview about daily functioning. Some of the other measures come from questionnaire data, where people answer questions about things like sensory experience, or their sensitivity to sensory experiences, or their own repetitive behaviors, or feelings about their abilities with social interactions.